Thank you so much for the kind introduction. And first, I would like to thank the organizers for the opportunity to present our research and technology in this forum. I'll be presenting our findings and aims really to leverage human virome to combat cancer, autoimmunity, and aging-related diseases. As you know, there are a vast and diverse community of viruses that infects humans, and these are collectively referred to as human virome. We are particularly interested in a group of viruses which we refer to as commensal viruses, and those are the ones that ubiquitously live in humans, if not all of us, most of us, and they do not cause any harm in immunocompetent individuals. We have discovered that compromised immunity to these viruses can in fact underlie the emergence of cancer, autoimmunity, and aging-associated diseases. And this discovery has really led, for the first time, to appreciation of the beneficial function of viruses as they regulate healthy immune response in the organs, and that in turn would maintain the health of that organ and tissue. And based on this concept, we developed a virome immunotherapy opportunity in which we have proof of concept for 2 viruses and discovery engine which would enable us to identify novel commensal viruses and indications for future development. So I'll first describe our Commensal HPV program, which is in the context of oncology, and describe what we have found and the potential of this as a therapeutic field, if you will. So HPV, or human papillomavirus, as all of you know, is a small double-stranded DNA virus which broadly colonizes epithelial cells in the skin and mucosal sites. And there are more than 200 types of these viruses exist in human. We have divided them into 5 genera of the virus. Everyone in this audience probably is familiar with alpha HPVs. The high-risk alpha HPVs belong to this genus of the virus, and those are the causes of cervical cancer and subset of oropharyngeal cancer. However, there's 4 other genera of the virus, beta, gamma, mu, and nu, which we have been studying, and these viruses are actually acquired by humans during early childhood. And they have a broad colonization pattern across the mucosal and skin sites, and they do not cause cancer. In context of immunosuppression, these viruses can generate wart, but in those of us who are healthy and immunocompetent adults, we don't really have any, if you will, symptoms or any harmful effects from these viruses. And that really initiated our interest in understanding the biology of these viruses and how the immune system regulates them. It led us to this discovery that the T-cell immunity against the virus within the skin not only prevents wart development, which is caused by the virus, but also cross-protects that tissue from carcinogen-induced cancers. In the case of the skin, that will be UV-induced carcinogenesis. And for the cancer-initiating cells then to be able to progress to form a skin cancer, they really have to get rid of the virus to evade that antiviral immunity. And that's really a very rare event, and we don't really see that in the clinic that much. What we see is that the compromised antiviral immunity that then leads to massive expansion and increased risk of cancer, namely in the elderly and also in the immunosuppressed individuals, for example, after organ transplantation. So this concept of antiviral immunity protecting against cancer was really started for us by looking at clinical samples of skin cancer. And as you can appreciate in this image on the slide, there is absence of HPV, this is beta HPV, viral activity within the cancer cells, even though you can detect the virus RNA, and these are, again, small red dots that are highlighted by the arrow, that are detectable in the normal skin adjacent to that cancer. In other words, the loss of HPV seems to facilitate the development of squamous cell cancer. In this case, we have quantified this across multiple samples, as you can see on the right, demonstrating that there is really a loss of virus as cancer develops. We took these clinical observations to the laboratory and tested the role of papillomavirus in both chemical and also UV-induced carcinogenesis models. And we find that colonization with papillomavirus in animals would protect them against skin cancer. This is perhaps easiest to appreciate by looking at the slide, on the images on the right side of the slide, which demonstrate mice that received papillomavirus, so they were first colonized by the virus and then exposed to UV carcinogenesis, versus those that were colonized by a virus-like particles or sham-infected and then subjected to UV carcinogenesis. The mice that carry the virus are much protected against cancer. So then, based on these discoveries, we really proposed this Commensal HPV immunotherapy platform, which boosts that T-cell response within the tissues, in this case skin, for better protection against warts and also protection against cancer. in the elderly and in immunosuppressed, and broadly speaking in humans that are at risk of developing skin cancers, which is a large population of patients that we see in the clinic. We took this concept of commensal HPV immunotherapy to the laboratory again and developed the mouse papillomavirus immunotherapy, which demonstrates 100% efficacy in eliminating lesions in mice. So this technology really is currently at the doorstep of clinical development, and we are really excited to take this into our patients, my own patients, who are at very high risk of developing deadly skin cancers due to the mostly compromised immune response to this virus. Now I'm going to switch gear and describe one of our other programs which relates to human cytomegalovirus, and this program is in the context of aging. I'm sure everybody is familiar with human cytomegalovirus, or CMV. This is a virus that all of us, if not all, most of us carry in our body. It's a very common, persistent virus in human population, and there has been decades of research on immune response to this virus, which has shown that many of us, especially as we age, we accumulate T cells in our body that are specific to detecting this virus. However, there hasn't been really an understanding of the functional role of those T cells in humans and how they really maintain that elderly, healthy individual that has massively expanded population of anti-CMV T cells. So we came into this field trying to really understand, again, a functional role for this axis between the virus, in this case HCMV, and the T cell response against it. And what we discovered was, in fact, as cells age or become senescent, they reactivate the virus within them, and that viral reactivation provides a very bona fide, if you will, antigenic tag for the T cells to recognize that senescent cells and eliminate it. And this is now very easy to conceptualize as a therapeutic, which is boosting that T cell response against those antigens of the CMV for better clearance of senescent cells. That relates to aging, aging-related diseases, and also other diseases that we know there's senescent cell accumulation as part of the pathogenesis of the disease. So overall, as I went through this, the virome immunotherapy technologies approaches in this context quite distinct from existing modalities. There's in fact no comparable modality in the field of virome, as we actually creating that field as we speak. But there are modalities that looking at vaccines, for example, in cancer. And what we are demonstrating here with commensal virus immunotherapy is a broad recruitment of adaptive immunity, T cells and B cells, for therapeutic benefit. And I would like to highlight that this is distinctly different from high-risk HPV vaccines that everybody is familiar with. Those vaccines are meant to activate B cells solely for production of antibody, prevention of infection with high-risk HPVs. Here we really would like to take benefit of the beneficial function of commensal HPVs, which are already in our tissues, and try to bring the T cells to better recognize those viruses and protect that colonized tissue from cancer. In terms of indications, as I alluded to, we are very active in context of cancer prevention and cancer therapy, but also aging, autoimmune diseases, and unfortunately I didn't get a chance to describe our finding in autoimmune disease and virome in that context. So key points with regard to virome therapeutics, we are really leveraging the natural biology of commensal viruses and immunity against them for therapeutic benefit. We have demonstrated both safety in animal models and human cells for these technologies that we have developed. And there has been a small-scale manufacturing process already established for HPV and our other programs. We have, as you can imagine, we have several patents secured around the virome technologies that we have developed in our laboratory. And we have proof of concept established for 2 programs, plus 2 pipeline projects that we are actively working on as well under the umbrella of virome therapeutics. Next steps for us, we have been successful in securing funding from MGB Amplify to establish CMC and take our HPV program to the pre-IND stage. And we are actively now establishing a new co and raising Series A to enable GMP to first-in-human trial for our HPV program, and also proof of concept to IND for other programs that we have. So in closing, I would like to restate that our big goal here, big aim here, is to harness the beneficial function of human virome to combat major diseases in human, including cancer and autoimmunity. And with that, I would like to thank you for your attention.